Okay, in this segment, I want to talk about a basic introduction to human genomics. Obviously, if we're thinking about pharmacogenomics, we need to understand pharmacology, but we also need to understand the human genome and the sources of variation. So let's start off with some basics. Humans are diploid, which means we really have two genomes in every cell, one from mom and one from dad. And it means that for every gene, in general, we will have two copies, the copy from mom and the copy from dad. A genome in general is 3 billion DNA bases. Remember the DNA bases, A, T, C, G. Genome, 3 billion. It's a big number, but it's not a huge number. You could store your genome as A, T, C, and G on a modern iPhone with very little problem. It would be like 3 gigabytes. It's divided up into 23 chromosomes. It's not very important for our discussions in general. You could think of this as a single 3 billion, but actually it's been divided up into the 21 autosomal chromosomes plus X plus Y. Great. Now, in those 3 billion bases are about 25,000 genes. I put genes in quotes because a lot of people know what genes are, but it's actually one of those things where you kind of say, I know it when I see it, but it's hard to define. So for our purposes, I'm going to define a gene. Let me get my notes. As a DNA sequence in the genome associated with a particular biological function. So if you think about genes that you might be familiar with, hopefully this definition is compatible with that. And if you use this definition, you will define about 25,000 genes in the genome. Interestingly, only about 2% of the genome is DNA encoding for this genes, which means 98% or so of the genome is non-coding and therefore involved in the control of gene expression or other important activities within the cell, but not coding for genes. So what does a gene look like, and how do we think about the genome? So this is not to scale, but a typical gene looks like this. Let me draw it, and then I will. A gene will typically have coding region, which I'm drawing as these 2D boxes, and which are going to code for amino acids, and we're going to call those exons. So this is exon 1, the beginning of the gene, exon 2, exon 3. But in the genome, the exons are separated by non-coding DNA called introns. So this is intron 1, intron 2. All the stuff to the left of the gene is going to be called intergenic DNA, literally between genes. And this is going to be intergenic as well. Now, I told you that 2% of the genome is genes. So that intergenic space could be huge, much bigger, way off of our whiteboard, way down the block in both directions, if you're talking really about 2% of the genome. So this is not to scale, as I've said. But these exons encode the amino acids of the protein that is encoded here in the gene. Now what happens is through, this is just a review of your biology, there is transcription by which messenger RNA is a copy of the genome, and there's actually a pre-message, it's called pre-mRNA, that includes the exons and the introns. So that's E's plus I's. But then there's a splicing step that removes the I's, removes the introns, and then you get a messenger RNA, which is kind of what you would expect, which are the 3 segments spliced together into a contiguous gene that can now be decoded through translation. As you notice, I am reviewing the central dogma of biology. Translation will turn this into an amino acid sequence. You will read 3 bases at a time, 3, 3, 3. Notice I've straddled an exon boundary here. That does happen. So you could have the first 2 bases on one exon, the last base of the codon that specifies amino acids in the next exon, etc., to get all of your amino acids, and then you get your protein structure, which will fold into some beautiful shape. And of course, the translation happens at the ribosome. So that's a quick review of how genes in DNA get transcribed to messenger RNA, first pre-mRNA, then spliced into the mRNA, messenger RNA. The messenger goes to the ribosome and through translation gets translated into a protein. Okay. 25,000 or so of those. Now, a couple of important concepts that we need, especially in order to understand things that will come up in pharmacogenomics, is we have this idea of gene homologs. Genes are homologs if they have a common ancestor. Life on Earth is about 4 billion years old. 4 times 10 to the 9th years, we've had life on Earth. For many of those years, we were just single-cell little amoeboids. But then eventually through evolution, mutation, and selection, multicellular organisms, mammals, fish, plants, the whole ball of wax. But what we see is that many genes share common ancestors because of this tree of life. And there's a couple of kinds of homology that I want to discuss. One kind are orthologs. So orthologs are 2 genes in different species that have the same function. 2 genes with same function in different species. These are very interesting because, for example, if you've identified a gene in yeast that has an ortholog in human, it is much easier to study yeast than humans. You're allowed to chop up yeast and kill them and study them, whereas that's generally not allowed for humans. Okay? So orthology is extremely useful for lots of biological experimentation. But for pharmacogenomics, it's not quite as useful because we're interested in human proteins. But what we do have is the idea of paralogs. Paralogs are 2 genes in the same organism. 2 genes in the same organism that are homologs, so they do have a common ancestor, but their functions have diverged. Functions have diverged. All right, so why am I telling you about all this? Well, it's very important for pharmacogenomics because what we see in evolution is sometimes there's a single gene, so there's a gene sitting in the genome, and something happens during the course of evolution that gives the organism 2 copies of the same gene. So 2 copies of the same gene means that they're paralogs. Now, but I said that the functions have diverged. On the day that this happens, these are identical copies and might be doing exactly an identical thing. They're still paralogs, but we don't usually get to observe that. What happens is, this had some critical function for life, and one of these guys has to continue doing that critical function. But the other guy is now free to evolve. Since this guy is taking care of business, if you will, this guy can evolve to do new functions. So this is key function x. This guy will do key function x. This guy can do optional new function y. This has happened in the human genome many, many times. So there are many gene families in the human genome which have the following characteristics. They all have very high sequence similarity. High sequence similarity. That's kind of how you detect that they're that they're paralogs. But they have diverse functions, divergent. And I'm sorry if this is too small. So divergent function. So I talked about, in other lectures, other segments, I talked about the ABCB1, the ABC transporters. They are all derived from a mother ABC from which they all became divergent paralogs with slightly different specificity for transporting different types of molecules. I talked about the cytochrome P450s, the CYPs, the phase 1 metabolizing enzymes. They also have a mother ancestor and now have all diverged and copied themselves in the genome to have slightly different functions. So it's important to remember that many of the key proteins in the human genome that are involved in pharmacogenomics, especially metabolism and PK, are members of these gene families. That does raise some issues in terms of challenges for sequencing or measuring the DNA genotypes of these genes, which we might discuss later. But it's very important to remember this idea of homologs, paralogs, and the ubiquity of paralogs in many of the families important for pharmacogenomics.